Good afternoon, and welcome to the last session of the day, covering an interesting topic, timely topic that seems to be populating every field in medicine nowadays. We are going to talk today about the role of the microbiome or microbiota in pancreatic diseases, pancreatitis and pancreatic cancer. We have three speakers, and Dr. Julia Miley, you all know her. She's a familiar face here in our association. We'll introduce our first speaker.
Thanks a million, Carlos. No need for introducing you. So we're happy to have Cemal Yazici from the University of Illinois. He's a clinical gastroenterologist doing ERCP and EUS service, so very much involved in pancreatitis care. To have him talk about modulating the fecal microbiota to influence severity of acute pancreatitis from mice to men. Cemal, we are looking very much forward to your talk.
Thank you. I would like to start by thanking APA Conference Committee for giving me the opportunity to present our work. I have no disclosures to make. I will briefly review gut permeability, bacterial translocation, and AP outcomes. We'll cover in vivo studies that focus on gut microbiota and acute pancreatitis, review some of the recent translational investigations, and then provide some preliminary data from our ongoing investigation. And then we'll end up with summary slide. So the role of gut microbiota is really well recognized in disease development, and it has been a common area of interest, especially in regards to acute pancreatitis risk and severity over the past decade. And microcirculatory injury and hypovolemia, we know, can impact gut permeability. And this can increase the bacterial translocation and subsequent development of local and systemic complications. We know that it's a major challenge, and especially for those who have moderately severe and severe acute pancreatitis, especially the ones with infected pancreatic necrosis do worse, and that underlines the importance of gut microbiota in AP outcomes. There are studies in the literature, but there are also limitations, such as lack of mechanistic explanations, inadequate phenotyping of AP patients, as well as applicability of these findings to general population. So we know of earlier in vivo studies show that probiotics reduce bacterial counts of potential pathogens in duodenum, as well as decrease the bacterial translocation to pancreas. They also improve survival in mice treated with AP prior to induction of acute pancreatitis. Similar studies also show that acute pancreatitis actually can impact gut microbiota, and that this can be reversed with introduction of probiotics prior to AP induction. And probiotics mice treated with probiotics have decreased total bacterial counts, less bacterial translocation to mesenteric lymph nodes, as well as decreased total pathology scores in examined pancreatic tissue. Similar changes have also been shown in hypertriglyceridemia-related acute necrotizing pancreatitis. Animals were, in one study, separated into two groups, those that are fed with normal lipid versus high lipid diet. And the most impact was seen at 48 hours. So investigators looked at it and showed significant decrease in alpha diversity in gut microbiome in each group. And also they were able to show that there were significant differences in relative abundance of phyla and genera in gut microbiota in these treatment groups. We also know that antibiotic treatment markedly decreased morphological damage in the form of necrosis in pancreas, and fecal microbiota transplantation actually re-aggravates this pancreatic injury. Similar studies have also shown that gut microbiota depletion by antibiotics elevated acute pancreatitis severity, and they have shown similar findings in germ-free mice as well. And another study replicated this finding, and again, it was shown in germ-free mice. So from here, we will transition and briefly talk about human subject studies, and a recent study that was published in 2015 also showed a similar effect. Those who had mild acute pancreatitis severe acute pancreatitis, they had decreased bacterial loads of pathogenic bacteria and increased loads of beneficial bacteria in the control group. And a recent study showed gut dysbiosis was also seen in acute pancreatitis patients. And if you look at the panel, you can see that those who have acute pancreatitis separate nicely from those healthy controls. And they were able to correlate predominant genera in the AP group with inflammatory markers in this study. And they further separated the acute pancreatitis patients into severe and mild and moderately severe group, and they were able to show that even the severity itself actually is associated with differential abundance of certain genera. And one of the important findings from this study was that they were able to show enhancement of the pathways for infectious diseases such as bacterial invasion of epithelial cells in acute pancreatitis patients compared to controls. So from here, I will introduce our study briefly. We know that also there are recent epidemiologic studies that showed that patients who consume diets high in red meat and low in fiber have increased acute pancreatitis risk. And we all know diet can modify gut microbiota composition. And the in vivo data, as well as observational studies, point to contribution of gut microbiota to acute pancreatitis risk and severity. And based on this, we developed a working hypothesis model, mainly focused on gut microbiota. So decreased diversity can result and it is associated with increased abundance of particle group of bacteria that we are interested in, sulfidogenic bacteria. This has been studied extensively in literature, not in pancreatitis. So they are known to produce hydrogen sulfide, which can have oxidative and inflammatory effects, and this can potentially increase acute pancreatitis risk. Also, gut microbiota, we know of modulate short-chain fatty acid composition, and they are crucial for gut integrity. And either of these mechanisms, or they are combined, can increase acute pancreatitis risk and severity and recurrence and subsequent development of AP-related exocrine and endocrine insufficiencies. So in our case control study, we recruited a total of 70 patients, 37 acute pancreatitis and 33 control subjects. They were very similar in regards to age, sex, education level, income level, BMI, waist circumference. They had similar intakes of statin use and NSAID use, as well as similar smoking. We served to a very diverse community, so we had higher percentage of Hispanics with acute pancreatitis in our AP group and higher percentage of African Americans in our control group. So that was pretty much the major difference in these two groups. The most common etiology for acute pancreatitis was alcohol, followed by gallstones, hypertriglyceridemia, idiopathic etiologies. Close to 25% of our patients had severe acute pancreatitis, and approximately 32% had prior history of acute pancreatitis. So we collected habitual dietary intake from these patients, and the major differences here were that acute pancreatitis patients had lower consumption of some of the beneficial polyunsaturated fatty acids. They also had lower consumption of omega-3, protein from seafood and fish, lower daily servings of fish and whole grains and vitamin D, and they had higher intake of alcohol. We collected stool specimens from our group, and we looked into gut microbiome diversity, and acute pancreatitis patients had significantly decreased gut microbiome diversity compared to controls, and this was more pronounced in those AP patients with diabetes. And the panel on the right side shows relative abundance of certain genera among these three groups. Also, the linear discriminant analysis identified differential abundance of genera associated with acute pancreatitis versus control studies. The one on the left side, the one with acute pancreatitis patients, and the bigger the circle, the more the significance is. So if you look into these specific genera, almost half of those genera on the acute pancreatitis group were identified as sulfidogenic bacteria, producers of hydrogenous sulfide. These include Veillonella, Klebsiella, Enterococcus, Actinomyces haemophilus, Rothia, Gamella, Morganella, and Prevotella. In opposition, the control group had higher abundance of bacteria that are involved in short-chain fatty acid production. We further did an LFC analysis, and it identified specific genera associated with acute pancreatitis status. These were Veillonella and Haemophilus, and 6 other bacteria were associated with control status. And if you look into panel B on the right side, you can see that there are distinct differences and associations among bacteria in the AP versus control groups. We also look into potential bacterial and metabolites. We checked serum short-chain fatty acid levels. We did not see any significant difference in butyrate or propionate level among 2 groups, but acute pancreatitis patients actually had significantly higher acetic acid levels compared to controls. And acetic acid is one of the end products of alcohol metabolism, and that makes sense if we think about a couple slides earlier. We were mentioning that alcohol was the most common etiology in our group. Also, those genera that were associated with acute pancreatitis, most of them also have been identified as acetic acid producers, some of them. So we think that clinically that fits well with the clinical data that we have. We also look into hydrogen sulfide levels. These are the metabolites of sulfidogenic bacteria. And it was significantly increased in moderately severe and severe acute pancreatitis group compared to other groups. Finally, we used a machine learning model and had an ROC curve of 96, ROC curve with 96% predictive ability to differentiate acute pancreatitis patients from controls. And it was based on the 24 taxa and also acetic acid levels. And the panel on the right side actually shows the importance of these 24 taxa based on the random forest model. So in summary, gut microbiota diversity is significantly decreasing acute pancreatitis. This has been shown in vivo models and also human subject studies. Alteration of gut microbiota composition appears to be more pronounced in those with severe acute pancreatitis. Probiotics and FMT have been shown to modulate the degree of pancreatic injury in animal models. Also, potential gut microbiota-related pathways have been involved in acute pancreatitis, and they are right now emerging in human subject studies. These include pathways involved in bacterial invasion of epithelial cells, production of inflammatory and metabolites by gut microbiota, such as hydrogen sulfide, and also alteration in production of beneficial microbial and metabolites, such as short-chain fatty acids. And also yesterday we had a talk about how gut microbiota might be related to immunological alterations, and perhaps this could be also one of the pathways. So we need a large multi-site prospective human subject studies. Those will help us better to understand the relationship between diet, gut microbiota, acute pancreatitis risk, and related endocrine and exocrine insufficiencies. And we hope that these larger studies can generate the data for development of new strategies for either dietary modification or modulation of gut microbiota and their metabolites to decrease acute pancreatitis risk and severity. I would like to acknowledge our collaborators, the KL2 program at UIC, the members of our lab, and also NIH for their support.
Thank you very much. The paper is open for discussion. But just stay here. I think it's much better to have it. Aliyah.
Dr. Yosic, a very nice talk. Maybe there's no answer yet for this question, but as you alluded, what is the chicken, what is the egg when it comes to microbiome? And the diet really impacts the microbiome. So do you anticipate that perhaps, with in countries where there is an increase, monosaturated fat consumption is higher, such as olive oil, or the fish consumption is higher, you would expect a different microbiome composition, therefore differences in acute pancreatitis and AP-related diabetes complications?
Yeah, that's a very good question. Thanks, Dr. Woods. So, yeah, the main point in the presentation was the modulation of the gut microbiota and how it relates to acute pancreatitis risk. It's true one can extrapolate that and try to generalize it, and it makes sense conceptually, but we know that also there are many other risk factors that can impact gut microbiota behind diet. So I think it will be really difficult to generalize, but perhaps having this replicated in a lot more larger data set that perhaps includes some subjects from those countries as well, they can help us better understand and sort the differences among the potential populations in these groups.
I had a technical question and question. How was this microbiota collected and stored? Were they rapidly frozen or kept at room temperature 4 degrees?
Yeah, so these patients, we recruited all of them from hospital. So both AP patients were recruited when they came to the hospital and admitted for acute pancreatitis episodes. And similarly, control patients, because we thought that gut microbiota composition may be impacted even with the hospitalization status. They also have been recruited from hospital, but they were patients admitted to hospital for treatment of other conditions not related to pancreatic diseases. We collected the first stool sample from these patients when they produced it in the hospital, and they were frozen directly and stored in -80 until the DNA was extracted from these stool samples and was processed for 16.
Were they stratified based on opioid use? Or
No, we haven't, and I listened to your talk and learned in regards to how also it may play a role in modulation of pain. So. But it wouldn't be too difficult. Our sample size is small, but right now we have up to 50 AP patients. I think it will be easy to just go into the electronic medical records and look into use of opioids during hospital, and if they reported pain, potential retrospectively, we can look into that as well.
Very nice talk. Thank you.
Thank you.
I have 2 questions. The first one has to do with your difference in the diabetics. Do you think that the difference that you saw was due to their gut motility causing the changes in the microbiome, or was it something to do with the pancreas itself?
Yeah, those are really good questions as well. So we know that gut motility is impacted in diabetics, so that could be an important reason, but also we want to do that subgroup analysis because diabetes itself is also associated with altered gut microbiome, diversity and also difference in gut microbiome. So it is really difficult for me to see if it is related to motility, but we try to control it, as best as we can by taking diabetes status into consideration. And this diabetes was existing diabetes at the time of acute pancreatitis. So.
And the second question I had, that similar to the opioid question, what about proton pump inhibitors, which we know alter the microbiome? Did you take that into consideration in your patients?
No, we haven't,
Okay.
but we should be easily.
You
We should be easily, access that information, and we can include that in our perhaps model adjustment.
Peter.
Very nice talk. 2 questions. How representative do you think the stool sample is of the gut microbiome, which is much more upstream, potentially coming from the small bowel, especially with the stasis, ileus that can develop in pancreatitis and other drugs that patients may be on, like opiates and PPIs? How representative is the stool of the upper GI tract?
That I'm not really sure if we can clearly say that they are similar. So these are all stool samples, and we know that there might be differences between the luminal stool samples and mucosa adherent microbiota, or perhaps the microbiota that can be isolated from lumen, such as duodenal juice. So I'm not really sure how well it represents the microbiome upstream in the upper GI tract.
The second question is in regards to dietary patterns. Usually dietary patterns are a lifetime habit. That's the way we cooked all our life. That's the way we will keep cooking all our life. So have you looked, or how do you conceive this, that the patient who comes in with pancreatitis, several months before he developed pancreatitis, was eating the same diet, and probably will go back to the same diet after he finishes the attack of pancreatitis and goes back home. So, because it's going to be the same diet, whether it was before or after the attack, how does that influence your conclusions about the role of the microbiome that you isolate at the time of the attack as being representative?
Yeah, that's a very good question as well. So the dietary data tool that we use looks into dietary intake over the past 3 months. So we thought that that will probably represent their habitual dietary intake. But it's a good question. Of course, if you alter your dietary intake, that's probably going to have a major impact in your gut microbiota composition. Most of these patients, of course, they go back into their communities, and unless they are advised on diet, they may start to go back and have a very similar diet, and that may have additional impacts. And that's one of the endpoints that we are trying to lead in the future, to see if change in diet following initial acute pancreatitis attack can actually prevent a recurrence of similar AP attacks.
Thank you very much, Dr. Azizi. That was a very interesting discussion and talk. Our next speaker is Dr. Cindy Nuzillet from the Curie Institute in Paris. She's going to be a virtual talk. She has published extensively in pancreatic cancer as well as on the microbiome, and comes with a very provocative title for her talk: Microbiome, Pancreatitis, and Pancreatic Cancer, a Continuum.
Good afternoon. I'm Cindy Nuzillet. I'm a gastroenterologist, and I'm the head of GI oncology at Curie Institute in Saint-Cloud, France. First, I would like to thank the organizers for inviting me to talk about microbiome and pancreatic cancer. It's a great honor and pleasure, and I apologize for being unable to attend physically the meeting with you tonight. So here are my disclosures. And this is the overview of the presentation. So first, I will present the data about the association between oral and gut bacteria and pancreatic cancer. Then we will focus on the very exciting data that have emerged in recent years about the role of intratumor microbiome in pancreatic cancer. And finally, I will discuss the clinical applications of these results with some ongoing work from our team. So first, to remind you that pancreatic cancer incidence is increasing worldwide, and particularly in Europe and in the US, where it's expected to become the second cause of cancer-related death by 2030. And there are huge discrepancies between countries and between ethnicities around the world, with a fourfold difference between lowest rates observed in Africa and highest rates observed in Europe and North America, while African Americans have the highest incidence of pancreatic cancer in the United States. And this increase in incidence and disparities are not fully explained by so-called classical risk factors of PDAC, namely aging, tobacco smoking, obesity, diabetes, chronic pancreatitis, and family history and genetic factors. Besides these well-known factors, periodontal disease has been explored as a risk factor of PDAC since the early 2000s. And in this meta-analysis of 8 studies looking at periodontal disease and endontulism as risk factor of pancreatic cancer, an association was found between these two conditions and pancreatic cancer risk, with a relative risk of 1.74 and 1.54. This was further confirmed by studies looking at the oral and salivary microbiome composition using sequencing of the 16S ribosomal RNA gene of the bacteria. This study revealed differences in the composition of the oral microbiome between cancer patients and healthy controls, with a decrease in Neisseria elongata and Streptococcus mitis in cancer patients, while there was an increase in Porphyromonas gingivalis and other periodontal pathogens in cancer patients. This was further supported by data from the EPIC observational cohort, showing higher levels of antibodies against Porphyromonas gingivalis in pre-diagnostic blood samples of PDAC cases compared to controls. And so this bacteria, Porphyromonas gingivalis, is the one for which we have the most abundant literature for association with PDAC risk. Regarding the gut microbiome, the number of articles is more limited, but we have data showing decreased microbial diversity in cancer patients and also qualitative differences with an increase in the ratio between Bacteroidetes and Firmicutes bacteria. Moreover, some changes in the oral and gut microbiome have been described in relation with some of the risk factors of PDAC and with some treatments such as antibiotics, PPI, chemotherapy, and metformin. So this alteration in oral or gut microbiome might directly promote PDAC development or be an intermediate mechanistic step between the risk factors and PDAC development. But in all these studies, we just have associations described, but no true mechanistic data. So we don't know if it's just a coexisting conditions or a consequence of PDAC rather than a causal relationship. If we move now to the intratumor microbiome, it's the field in which there have been the most exciting data emerging in the recent years. So the pancreas has been considered to be a sterile organ for a very long time. And in 2017, Geller et al., so from a consortium of teams working in Israel and in the US, described for the first time that bacteria were present in pancreatic tumors. They detected bacteria in 76% of PDAC compared with only 15% of normal tissue. And they showed that gamma proteobacteria was the most abundant class of bacteria in these tumors, which was further confirmed by work from other teams. Further, 3 years later, they showed that these bacteria were found predominantly intracellularly, so in the cytoplasm of the cells, either the immune cells, including macrophages, but also in cancer cells. And they compared the microbial composition of different tumors, including pancreatic cancer, and they showed that PDAC had a very distinct bacterial composition compared with other cancers. Bacteria can also be found in cysts, in pancreatic cystic lesions, and particularly in IPMN, with this work from Geiser et al. Showing an increased abundance of 16S DNA copy number in patients with IPMN compared to non-IPMN patients, along with an increase in the levels of IL-1 beta. And they showed that the copy number increased with the grade of the dysplasia, with higher levels of 60-days DNA copy in patients with high-grade IPMN compared to low-grade IPMN. Where do these bacteria come from? So the hypothesis is that they can be translocated from the duodenum, even if we don't have clear mechanistic demonstration of that. This is based on the study of human samples from different parts of the gut compared with the pancreas, showing that the duodenum samples segregate together with the pancreatic samples in PCR analysis. And in preclinical model, when we use mouse oral gavage with fluorescent bacteria, we can find the bacteria in the pancreas of the mice at sacrifice. So this is also an argument in favor of the translocation from the gut. Now that we know that bacteria are present in pancreatic tumors, what are the impacts on patient outcome? So this is a very important paper by Riquelme et al., in which they compared the intratumor microbiota of pancreatic cancer patients with either a short-term survival or long-term survival. And they described qualitative difference in terms of microbiome composition between short-term and long-term survivals, with more an higher level of diversity in patients with long-term survival compared to patients with short-term survival. And they developed an intratumor microbiome signature, which was highly predictive of long survival and validated in 2 independent cohorts. Now, the question of the talk is there a continuum between pancreatitis and PDAC in which bacteria could be involved? So first, I wanted to remind that the continuum between pancreatitis and PDAC is well established in mouse model, because we know that in genetically engineering mouse model in which the pancreatic acinar cells carry only KRAS mutation, it takes several months for them to develop preneoplastic panning lesions. But if we want to accelerate the carcinogenesis, we have to add an inflammation trigger. So pancreatitis, either, for example, using high-fat diet or cerulane. And in this case, it will activate the oncogenic KRAS signaling by the activation of the surrounding microenvironment, so including the activation of protumoral immune cells and also of fibroblasts. But in human, the association is less clear, because we know that chronic pancreatitis is indeed a risk factor of PDAC, but the absolute risk of pancreatic cancer in patients having chronic pancreatitis remains low, and this is why there is no organized PDAC screening in this population. And the majority of PDAC develop without underlying pancreatitis. Nevertheless, we know that the tumor microenvironment play a major role in pancreatic cancer. The stroma represents the major part of the tumor volume, and the stroma is composed of extracellular matrix and immune cell and fibroblast. And now there is a new partner in this stroma, which are the bacteria from the intratumor microbiota. And these bacteria have been shown to play a role in pancreatic carcinogenesis from the very first step of the carcinogenesis. Indeed, using a KC mouse, Puchalka et al. Showed that if they use axenic mouse, so which are mouse without any bacteria in their gut, and they repopulate the gut with feces from either wild-type mice or tumor-bearing KPC mouse, they show that KPC feces are able to accelerate the formation of preneoplastic lesion. And they show that the ablation of bacteria from the gut is associated with an increase in CD3 positive, so T cells in the microenvironment, possibly involved in the control of the development of these preneoplastic lesions. Moreover, different teams use different strategies to remove the bacteria to study the effect of these bacteria on pancreatic cancer progression. So Sethi et al. Use antibiotics in a model of subcutaneous KPC cells grafted in immunocompetent mouse, and they showed that antibiotics were able to slow the progression, the growth of the tumor in these mice, and the effect was abrogated when they use RAG1-KO mouse, so mouse without mature lymphocytes. This was also supported by these results, showing an increase in cytotoxic T cells in the tumors of mice treated by antibiotic compared to control mice. Similarly, Puchalka used an orthotopic KPC graft, so in the pancreas of immunocompetent mice, and they showed that antibiotic reduced the tumor growth, and that this was mediated by CD4 and CD8 cells, because when you co-treated the mouse with these antibodies, you lose the effect induced by the antibiotic. They showed in terms of immune infiltrates that antibiotic increased infiltrates by antitumoral T cells and M1 macrophages, and decreased infiltrate by MDSC and M2 macrophages, which are pro-tumoral, and that these effects were TLR mediated. Finally, Riquelme et al. Used another strategy, fecal transplantation, to study the effect of the microbiota in an orthotopic KPC graft model. So they treated these mice with antibiotic to remove the bacteria from their gut, and then they repopulated the gut with feces from either short-term survivors or long-term survivors with no evidence of disease. And they show that the feces from long-term survivors were able to reduce the tumor growth, and this was associated with an increase in cytotoxic T cells and a decrease in T regulatory T cells and in MDSCs. And the effect was mediated by CD8 because when they co-treated the mice with antibodies against CD8, the anti-tumoral effect was lost. Interestingly, the same differences in terms of composition, prognostic impact, and pro-tumorigenic role have been described as well for fungi. So besides the microbiota, we have also the microbiome. And in this paper from Acute et al., they demonstrated that fungi were also involved in PDAC carcinogenesis. So these are very interesting mechanistic effects, limited to mouse, but it showed how bacteria can interact with immune cells to promote tumor progression. And also something very clinically relevant is that bacteria have been involved in chemoresistance. And in 2017, Geller et al. Showed that among the bacteria present in pancreatic tumor, we can find Klebsiella pneumoniae and E. coli K12 bacteria, which harbor long form of CDA, so the enzyme responsible for the degradation of gemcitabine. And indeed, these bacteria can degrade gemcitabine in vitro. And in a mouse model using a colorectal cell line graft, they show that when they remove the CDA activity from the bacteria, or when they use antibiotics against these bacteria, they can induce an anti-tumor effect in combination with gemcitabine. So antibiotics could be able to reverse the resistance to gemcitabine induced by CDA from these bacteria. In patients, we have several observational studies in different regions of the world showing that patients with metastatic PDAC who receive gemcitabine first-line chemotherapy and for any reason antibiotic at the same time have longer progression-free and overall survival compared to patients who don't receive antibiotic. Similar findings I've been also reporting in the adjuvant setting with patients who have Klebsiella pneumoniae in their bile have shorter survival compared to patients without these bacteria in their bile. And this survival effect can be reversed when they receive quinolones after their surgery. There are also other effects of bacteria on the pharmacokinetic of chemotherapy. For example, they are involved in the metabolism of irinotecan in the gut, and they can modulate the toxicity of this chemotherapy. And this is summarized in this very interesting review. So here you have the summary of the different effects of the bacteria in pancreatic cancer development, and mainly it's indeed mediated by pancreatic inflammation. So here is the link with pancreatitis and by the induction of immune suppression. But also we have this interesting data about their role in therapeutic resistance. So now, what are the clinical applications from these results? So bacteria are emerging as a new source of biomarkers and therapeutic interventions for PDAC patients. Several observational studies are ongoing to study the diagnostic, the prognostic, and the predictive role of bacteria in pancreatic cancer and other GI cancers. In our group, we are going to study the predictive value of intratumor microbiota for response to a vaccine in our TEDOPAM Prodige 63 study, which is a maintenance study in which patients receive either chemotherapy alone or combined with a TEDOP vaccine after FOLFIRINOX induction chemotherapy. And so we have a translational program in this trial on the blood and on the tumor, in which we will study the bacterial composition as a predictive biomarker for immune therapy. Moreover, we know that the microenvironment of PDAC is heterogeneous, and there are different kinds of immune cells, but also of fibroblasts, and there can be a spatial heterogeneity with some area with specific CAF and immune cell composition. And we hypothesize that these specific areas can be related to specific local microbiome composition. And we will explore this question using the ancillary material from the PRODIGE 24 study, which was a large phase 3 study in the adjuvant setting comparing FOLFIRINOX to gemcitabine. And we have access to the resected tumor samples from this patient. And we will perform both an NGS analysis of the bacterial composition along with a multiplex immunofluorescence analysis of the CAF subtype. And we will study the correlation between these two analyses to study the interaction between bacteria and CAF subtypes. Regarding the intervention, several trials as well are ongoing using either fecal microbial transplantation or antibiotics, but mainly in cancers other than pancreatic cancer. In pancreatic cancer in France, we think that it's time to move to prospective trial, and we are currently opening the antibiotic trial in which patients with metastatic PDAC will receive gemcitabine-based first-line chemotherapy are randomized between either a placebo or ciprofloxacin antibiotic with the aim to remove the CDA-producing bacteria responsible for gemcitabine resistance. So to conclude, recent discoveries about the presence and biological roles of intratumor bacteria in pancreatic cancer have opened a new avenue in research. These studies are mainly based on the detection of 16S ribosomal RNA, and the mechanistic results are still limited. So we need to develop more comprehensive approaches to study the microbiome and to capture their functional effect, and also to develop innovative therapeutic strategies to target selectively the bacteria of interest in these tumors. Nevertheless, this field is moving quickly, and we can expect that clinical applications based on bacteria manipulation will be generated in the very next future. So I thank you for your attention, and I will be happy to answer your question.
Dr. Nuzelet, this was a fascinating talk. I think you've opened a lot of doors, and I think this is obviously an exciting field, and I can predict that the next 10 years is going to bring a lot of answers to this. We have questions here. I want to just bring one simple one to you. So cancer of the pancreas is more common in the head of the pancreas than it is in the body and tail. Do we have the same prevalence of bacteria in tumors in the head of the pancreas that we have in the body and tail of the pancreas? Of course, the head being in close contact with the bile and reflux into the pancreatic duct.
Yes, thank you. This is a very interesting question. Indeed, we can expect, as I said, as bacteria is translocated from the duodenum, as the pancreatic head is closer to the duodenum, we could expect that the bacterial composition and abundance will be different between the head and the tail of the pancreas. We don't have so far a true documentation for this hypothesis, and that's why in the PRODIGE 24 trial, we'll have 300 samples, both from patients resected from tumor from the head and from the tail of the pancreas. And so we will have this information of bacterial composition, and I hope we will be able to provide some information, more detailed information about this.
Thank you. Dr. Little?
A very interesting talk. I was wondering how the bacteria get to the tumor or the site where the tumor begins. Do they swim up the pancreatic duct, and does the tumor environment is it particularly hospitable to the bacteria? And then in a related question, if that's true, do you see bacteria also in tumor metastases?
So the question of tumor metastasis in pancreatic cancer, we don't have data, but we have data in colorectal cancer, for example, where it has been described that primary tumors that are colonized by Fusobacterium we can find the Fusobacterium as well in the metastasis. So it's something very interesting. We have this impression that bacteria are following tumor cells in the metastasis and also in PDX model in colorectal cancer. In pancreatic cancer, we don't have such data so far. Regarding the way the bacteria get to the tumor, it's something also very intriguing because we see that in patients with poor prognosis tumor, the diversity is lower, but there are some specific species that seems to be attracted to the tumor. And we don't know yet by which mechanism these bacteria are attracted by tumor cells in their microenvironment, and they probably have a specific pro-tumor role because they are selectively attracted in poor prognosis tumors. So that's something that needs to be studied, but so far the mechanistic studies remain very limited.
A question here to the left.
Here, I have two questions. First thing is, I saw the paper that you said is you use the probiotics to treat the AP, the mouse, it will promote prolong the survival. But in the pancreatic cancer, the mouse, you use antibiotics, the cocktail, to suppress the tumor. Have you ever think about when we try to translate this antibiotics treatment into the human pancreatic cancer patient? Is that feasible or doable? How can we continue to treat the patient always with antibiotics, where it also affects the health of the patient?
Yes, it's true that this question of the balance between positive and negative effects of antibiotics is truly something that is unknown. It's very intriguing that in all other tumors, antibiotics are associated with a poorer prognosis and shorter survival in patients. But we have these different observational studies from different areas in the world showing consistent improvement of survival in patients receiving antibiotics with gemcitabine together. So with this hypothesis that it can modulate CDA. When we developed the antibiopac trial, we had a long discussion with infectiologists because indeed our fear was to select bacterial resistance and to have a bad effect on the microbial environment of the patient and on the hospital. And so what we decided is to give short course antibiotics. So the patient receives only 2 days of antibiotics at each infusion of chemo. So it's a short antibiotic in order to limit the risk of resistance acquisition in the gut of the patient and in the whole system. It's something that we are going also to study in this trial because we will look for emergence of resistant bacteria. But indeed, we do think that without a prospective study, we will not be able to answer this question. So we don't know what will be the result. Maybe it will be a negative trial because we will have more deleterious effect of the antibiotic on the gut of the patient, or maybe we will be able to confirm the positive effect. But I think we will not be able to move forward without this prospective trial.
My second question is, sorry, when we talk about the microbiota, the bacteria in the gut or in the pancreas, we talk about immunosuppression, immunoactivation. Have you ever considered maybe the bacteria have some metabolism, maybe also like either we can study it more specific in the cancer or something?
Yes, it's possible that there is a kind of symbiosis, metabolic symbiosis between the bacteria and the tumor cells. Also, the hypothesis may be that bacteria are selected by specific metabolic microenvironment. For example, we can think that in areas that are more hypoxic, you will have different bacterial composition between in this area compared to areas that are better vascularized in the tumors. So far, we don't know if there are causal relationship and a true, let's say, partnership between the bacteria and the tumor cells, or if they are just passive associations. So that's why we need to move forward with a preclinical model as well, but it's difficult to study these just very focused micro areas in the tumor, including the intratumor heterogeneity, even in preclinical models.
Time for one last brief question.
Okay, I don't know if it's brief. It's Daria Esterhazy from the University of Chicago. I have a question what your thoughts are on what's the chicken and the egg, because actually pancreas has very strong antimicrobial defenses normally. So maybe this duct reflex is always happening. It's just once you have reached a certain stage of dysfunction, you cannot fight it anymore. So have you thought about, for example, measuring antimicrobial peptides in even that microniche, to see whether that could actually permit certain microbes to flourish? In that case, it would actually be a role later in the pancreas rather than initiating it.
So the data we have to support that it's the bacteria inducing the tumorigenesis is data from mice using germ-free mice, showing that in a germ-free setting, the panning lesions take a very long time to develop, and you can accelerate their development when you have a gut with bacteria in the gut of the mice. So I think this is the data supporting that it's truly the bacteria promoting the inflammation and then the tumor preneoplastic lesions and then progression. But it's true that this relationship, this causal relationship is very weak, and we have no evidence in human for this. So that's why I think we need to move to prospective clinical trials to try to get stronger evidence.
Okay, thank you.
Thank you.
Thank you very much. We really could have gone on for another 10 to 15 questions. Unfortunately, we have to stop here. Thanks a lot for your presentation. Sorry for not having you here. And we move on to our next speaker, who also will present virtual, and that is Ekin Demir from the Technical University in Munich. Ekin has spent a lot of his time on investigating the role of pain development in chronic pancreatitis and pancreatic cancer, and he now will show us the association between pain in pancreatic cancer and the microbiota. Ekin, we are very proud that you will present here today, and hopefully we can get your movie or recording.
Good afternoon, dear colleagues. My name is Ekin Demir. I'm from the Department of Surgery at the Technical University of Munich in Germany. And in this session, I'm very pleased to hold this presentation about how gut microbiota affect disorders of the enteric nervous system. Although on the virtual, I hope that I will be able to give you the key messages related to how gut microbiota can affect several diseases of the human organism through its effects on the enteric nervous system. I think that for understanding the effects of gut microbiota on the enteric nervous system, we should have a good knowledge of the anatomy and physiology of the enteric nervous system. I'm sure that you have several times heard that the ENS is referred to as a second brain. This is in part related to the fact that the number of neurons in the enteric nervous system is around 400 to 600 million, which is actually a greater number than found in the spinal cord. So this huge network consists of thousands of small ganglia distributed from the upper esophagus to the very low part of the body, to the anal sphincter. So embryologically, the enteric nervous system is derived from the neural crest, and it's important to know that development of the enteric nervous system is not completed upon birth, but it actually continues in the postnatal phase, in particular in the first 3 months after birth. So the functional maturation is completed postnatally, and we know that this happens under the influence of the gut microbiota. So already in this very early phase, there is a profound and key interaction of gut microbiota with the enteric nervous system. The main functions of enteric nervous system are probably best known by you as well, but these include, of course, the secretion, the control of secretion, exchange of fluids across the epithelium, but also control of the barrier function at the gut wall. The enteric nervous system also controls local blood flow and the immune system, and it's also very influential in the control of the gut motility. So the enteric nervous system, like other neural networks, is composed of enteric glial cells and enteric neurons, And I'm sure that you can remember from the anatomy that there are two main types of plexus, neural networks in the enteric nervous system. That is the myenteric plexus and the Auerbach's plexus, and also the submucous plexus, the Meissner plexus. But these are only two of the plexus that are found in the enteric nervous system. In fact, the total number of plexus that are found in the enteric nervous system is around 12. So there are actually 12 layers of neural networks that are diffusely distributed through the intestinal wall. And the cells that generate electrical impulses and thus initiate peristalsis are not the enteric neurons, but it's rather the interstitial cells of Cajals, also known as ICCs, that are under the control of the enteric nervous system. So when we look at the types of neurons that are found in the enteric nervous system, there are four types of neurons, we can say. So there are the afferent neurons, there are the efferent neurons, there are the interneurons, and the motor neurons. Among these, the so-called intrinsic primary afferent neurons are very important. They're also known as IPANs because they detect mechanical or chemical stimuli like presence of food, mechanical distortion, but also signals from the gut microbiota directly from the lumen, because their synapses are found already in the epithelial mucosal layer, and they relay this information to the rest of the enteric nervous system. There are two types of interneurons, like ascending and descending interneurons. They mediate the communication between afferent and efferent neurons. And then there are these motor neurons that are responsible for the motility, for controlling the motility, which is why they innervate the smooth muscle cells and also those muscles in the muscularis mucosal layer. There are some characteristic neurotransmitters for excitatory and inhibitory motor neurons, among which nitric oxide is the classical inhibitory neurotransmitter released by the motor neurons. So if you look at this illustration, you can actually clearly see in the middle that this IPAN, the intrinsic primary afferent neuron, gets the information from the gut lumen, and that transmits this information to the interneurons on the one hand, but also to the ascending or descending motor neurons, so resulting in an enteric reflex. So these neurons, these IPANs, as we will see later, also have a key role in the transmission of the signals from the gut microbiota. And all this that I said, explained to you until now, represent the so-called intrinsic innervation of the gut wall. But there's also the extrinsic innervation. So presence of an intrinsic and extrinsic innervation is typical for any GI organ. And the extrinsic innervation is derived from neurons that are outside the guts. So these are the so-called either autonomic nerves, the vagal nerve in the nerve endings and branches, and the spinal afferents. So the autonomic nerves are the efferent nerves, the efferent nerve fibers. These are, as per anatomy, either the sympathetic or parasympathetic efferences. And the afferent nerve fibers are derived from the vagus nerve or from the spinal cord from the dorsal root ganglia. And when you look at this illustration, you can see that all these neurons, be it afferent or efferent, all synapse on the neurons that are present in the enteric nervous system. So they can also modulate the function of the enteric neurons on the one hand, but they can also get the signals from the gut wall in addition to the intrinsic primary afferent neurons. So once this information is received by these neurons, it's conveyed to the brain, it's processed there, and the efferent fibers signal back to the gut wall, resulting also in a large reflex loop. So you have probably heard several times about the brain-gut axis, but to be precise, we should refer to this as a gut-brain-gut axis or gut-brain-gut loop. So this illustration should show that once an information is transported from the intestinal wall, let's say to the nodose ganglia or the vagal afferents, this is then transmitted to the brainstem, to the medulla oblongata, where it's processed in the dorsal nuclei there, and then this is conveyed through the efferent fibers, like parasympathetic efferent fibers, back to the gut wall and there to the enteric neurons. So this is the anatomical correlate of this large loop between the gut wall and the brainstem back to the gut wall. One of the key discoveries in this field has been the discovery of cells that are part of the intestinal epithelium, but have direct synapses with the primary afferents, primary extrinsic afferent derived from the vagus nerve. The enteroendocrine cells actually have synapses called neuropods that have a direct connection with primary afferent fibers derived from the vagus nerve. So this means that once a signal or information is received by the enteroendocrine cells, this can directly be further transferred to the vagal afferents, and vagus nerve then transfers this information to the nodus ganglia and, of course, eventually to the brain. So this study is key in terms of understanding the fact that an information or a signal that is sensed in the lumen of the gut can be anatomically directly transmitted to the brain due to the presence of this wiring between the intestinal lumen and the brain. So I think it's a very fascinating finding, and this finding also draws our attention to the potential role of gut microbiota in this context, because all the signals that are derived from the gut microbiota can be sensed by several components of the enteric nervous system. So the gut microbiota secrete factors that pass through the intestinal epithelium. So this is the first prerequisite that is fulfilled. And once they pass there, they can be perceived by the intrinsic primary afferent neurons that are part of the enteric nervous system, but also of the afferent fibers that are derived from the vagus nerve or from the dorsal root ganglia. And then, so these microbiota can thereby participate in the gut-brain-gut axis, gut-brain-gut loop that I previously mentioned to you. So we heard a lot in the session about microbiota in the GI tract. I will not go into the detail in this context, but it's important to know that intrinsic factors, so the organism or patient-specific factors, impact gut microbiota composition, but also external factors, and probably also diet is the factor that is more influential than, for example, the genetic factors of the patient. So how do microbiota communicate with the enteric nervous system? It communicates through so-called bioactive factors, and in literature you will find the term enterocytes. There are three types of enterocytes that need to be mentioned. So there are the short-chain fatty acids, the neurotransmitters, and the gaseous factors that are produced by bacteria for their communication with enteric neurons. So short-chain fatty acids are a result of the microbial fermentation of non-digestible carbohydrates like starch. And the typical examples for these SCFAs are acetate, butyrate, and propionate. And as you can see here, these factors are already used by the host, for example, for generating long-chain fatty acids to be used as a primary source of energy for colonic cells, but also in the gluconeogenesis. And these fatty acids can also bind and activate a certain class of G protein coupled receptors, GPR receptors, that are present on enteric cells, especially on the enteroendocrine cells that I've shown to you to synapse with vagal afferents. So these SCFAs are key messengers between gut microbiota and host, but also between gut microbiota and the enteric nervous system. And one fascinating fact is that gut microbiota can also produce neurotransmitters. In fact, several well-known neurotransmitters like serotonin, dopamine, norepinephrine, GABA, nitric oxide, acetylcholine, and histamine are all produced by gut microbiota. There are some species that specifically produce certain neurotransmitters like Clostridium sporogenes that can produce serotonin, which then results in a depletion of serotonin from the enteric nervous system. There are species like Lactobacilli or bifidobacteria that produce the inhibitory neurotransmitter GABA, and there are also bacteria that produce the nitric oxide, which, as I mentioned to you, is a typical inhibitory neurotransmitter for motor neurons. So all these neurotransmitters can cross the epithelium and modify gut function, including the function of the enteric nervous system. So the gut bacteria can communicate with the enteric nervous system either directly or indirectly. Directly via binding of these factors to certain receptors, but also of factors like LPS on the toll-like receptors. Indeed, mice that are deficient in toll-like receptor 4 have decreased transit in vivo in the colon, and they have changes in the neural numbers and colonic and ileal enteric neurons. And the microbiota are also necessary for enteric glia development because germ-free mice have reduced numbers of enteric glia cells, and when you restore the gut microbiota, this deficiency can be rescued. And also the activity of the intrinsic primary afferent neurons is different when in germ-free mice. They have a decreased excitability, which is again reversible. So due to the fact that these IPANs have axons that extend to the mucosa, they can directly sense bacterial factors. And indirectly, this interaction happens due to the presence of toll-like receptors on immune cells. So when, let's say, LPS is released from the gut microbiota, this can bind to TLRs also on immune cells. And the cytokines that are released by these immune cells can impact on the enteric nervous system. But also enteroendocrine cells that are part of the epithelium and have the synapse with vagal efferents produce some peptides that can activate certain, let's say, receptors like GPR receptors, but also the short-chain fatty acids can also activate those receptors and lead to a release of hormones like GLP-1 and PYY from these enteroendocrine cells. And these hormones in turn bind to their receptors on the enteric neurons. And activation of these receptors then results in the release of enteric nitric oxide, which is an inhibitor neurotransmitter, resulting in, for example, in gastric emptying relaxation. So it is now well known that enteric nervous system is a key player in the pathogenesis of several diseases. So this has been beautifully shown in the context of some central neurological diseases, like Alzheimer's, Parkinson's, or multiple sclerosis. We know that the neuronal alterations that are found in these diseases can equally be found in the enteric neurons. We don't know whether this disease really starts in the gut or in the brain. So this is currently a matter of debate and investigation. But modifying, let's say, the gut microbiota and enteric nervous system function also results in an improvement of neurological symptoms in the mouse models, let's say animal models of these diseases. But also outside of the central nervous system, like in cancer and diabetes, there are profound changes found in intrinsic neurons of the gut wall or also of the pancreas. So what do we know about the interaction of pancreas, enteric nervous system, and the microbiota? In fact, very little about the nature of this trilateral interaction. There are some microbiota-derived factors like enkephalin, for example, or 12-HTE that can control duodenal contraction, glucose uptake, and thus attenuate hyperglycemia. But this is nearly more or less all we know about this trilateral interaction. But we should not forget that the pancreas also has an intrinsic and extrinsic innervation, just like the gut. And the intrinsic innervation is found around Langerhans islets. So if you look at these confocal microscope images, around every islet in a human pancreas, you will find a ganglion. And these ganglia are very influential and important for controlling the endocrine function. And if you look at, let's say, lower resolution images, you will see a huge neuronal network that surrounds the intrapancreatic islets. And if you look at this image of a human pancreas where all neuronal fibers are labeled, I think it's a very impressive image showing the extent of dense innervation found in the pancreas. So in such a context, to me, it's no surprise that there has to be a profound interaction between the intrinsic, let's say, neuronal network of the pancreas that is very closely related to the enteric nervous system, microbiota, and the pancreatic function. So the key messages that I want to give you are that the enteric nervous system is a key player in disease pathogenesis because it's a major modulator of gut barrier function and enteric homeostasis. So the role of the enteric nervous system in human health and disease, including pancreatic disease, has been largely neglected. I think this also holds true for pancreatic cancer, diabetes, and pancreatitis. Interfering with this interaction between enteric nervous system and microbiota can influence disease pathogenesis and progression because this has already been shown in animals' disease of neurological disease like Alzheimer's or Parkinson's disease. And when interfering with microbiota for treating pancreatic diseases, I think we should also consider the effects and the alterations on the enteric nervous system, which is equally present in the pancreas. So I think in this field, there is yet a lot to discover, and it will remain a fascinating field in the coming years. I thank you all for your attention. I'm very happy to take your questions.
Thank you very much, Ekin. The paper is open for discussion. So maybe I can start. I mean, it's an intriguing story. My question would be, do we know anything about the amount of, say, neurotransmitters produced by bacterial cells? Do we reach a threshold where we really see signaling, or is the amount so low that it's just a, say, neglectable finding?
I think this is a very intriguing question, as you say, Julia, because I think it's quite difficult to distinguish between the amounts of, let's say, these very small molecules like nitric oxide derived from bacteria as composed to those derived from the enteric nervous system. And when we also consider that this exchange of these factors is actually happening at a very low scale, at a very low resolution. So it's a very focal, let's say, events that are being exchanged between the gut microbiota and the enteric nervous system. I think this is technically quite difficult to measure and let's say compare the amount of these factors derived from microbiota versus the enteric nervous system. But I think what we should always keep in mind is that the human body contains more microorganisms than it contains cells. So the amount of, let's say, any type of factor that is derived from these microorganisms can therefore always be substantial and deciding in all these biological processes, I guess.
Fred.
So first, that was a wonderful overview. Thank you for putting it together and being up so late at night to answer our questions. The comments have sort of been unidirectional, the effects of the microbiome on the enteric nervous system. And certainly products of our enteric nervous system appear in the gut lumen, and secondary products, their connections to secretion are in the gut lumen. So to what extent does our enteric nervous system modify the microbiome?
Excellent question. I think related to this, we know even less, I guess. So we have disease models that are usually, let's say, central nervous system disease models, like, for example, multiple sclerosis models, where people are performed by microbiota transfers and then controlled for, let's say, alterations in the central nervous system upon change of this microbiome. But when we look at these models, improvement of the symptoms in these animals is always accompanied by a change in the microbiota as well when you looked at it longitudinally. But we don't know, of course, whether this is a direct effect of the enteric nervous system on the local microbiota. So I think this interaction between the gut, the enteric nervous system in between, and the central nervous system is so complex that it is difficult to infer that there is an effect of the enteric nervous system on the gut microbiota because of the, this loop suggests that all these alterations that we see in this model might actually be also caused by changes in the central nervous system. So the level of evidence does not support any, let's say, claim saying that the enteric nervous system also changes gut microbiota composition. I think there is definitely a lack of evidence related to this. But I think this is a highly interesting question.
Thank you.
I just wonder if you study the presence of hexametonium and see a change in the microenvironment.
We have not done this. Yeah, we have not done this yet. But I know that other groups, I'm not sure if they published it, but they did this previously, and they have indeed seen a change in the microbiota composition in different parts of the intestine upon hexametonium application.
Thank you for staying up to talk to us. I'm Jill Smith from Georgetown. I was intrigued, if I understood you correctly, that some of these bacteria secrete neuropeptides and things like that. So what happens to people taking SSRIs to the serotonin secreting bacteria? Are they affected at all? And could we use that to modulate these bacteria?
Yeah. So serotonin, in fact, is a special, I would say, neurotransmitter that mediates, let's say, the effect between the bacteria and the neurons, but also between the epithelium and the neurons. So the gut epithelium itself is also secreting serotonin, and it directly acts upon the submucosal neurons. So there are some studies, correlative studies, that have shown, let's say, differences in the enteric neuronal activity in these plexus, in the enteric plexus, like the submucous plexus of patients who have undergone, let's say, colonic or ileal biopsies, taking SSRIs, and there are indeed changes in neuronal excitability of these patients. So this is something that we know, but to my knowledge, beyond this, there is not much known.
Thank you.
Again, can I ask you one last question? I mean, to add a layer of complexity, and keeping in mind that in pancreatic cancer, you see those neurons, and having learned from Cindy Nuzile that you will find LPS and bacteria inside the pancreas, have you ever looked whether there's a special, say, whether the bacteria can be found in direct vicinity of nerves, neural cells?
Yeah, unfortunately, we have not looked at it yet. So I have to disappoint you regarding this excellent point, but something indeed that was going through my mind when I prepared this presentation, but we have not looked at it yet.
Thank you very much for staying up that late and giving us a wonderful talk. And I think it's time to conclude this session. It has been a very interesting session and a wonderful day. And there's just one, and I really like to thank all the three speakers for excellent talks. And there's just one small announcement, and this is that this evening's reception, due to the weather, has been moved to the Mona Lisa Ballroom, which is off the main lobby on the Nobu side, and it will start at 7 p.m. And we're very much looking forward to see you all there. Thanks a lot for your patience and your attention.